me well?
Yeah, yeah. Yes.
Wonderful. Well, thank you very much for inviting me. It's my tremendous pleasure to speak to you. I enjoyed very much the preceding talks, and I'm glad that I can reach out from Chapel Hill, North Carolina, despite pandemics. First of all, I want to make sure that I claim my conflict of interest. I've studied a few companies. We have one company joined with Robert Luxenhofer and Rainer Jordan called Delacqua, which is in particular relevant to what I'm going to show you. I will be talking about polymeric micelles mainly in the context of, I would say, pharmaceutical performance rather than polymer science. Nevertheless, I hope you will appreciate the relationship between the performance and polymer science. And of course, I want to start from the disease, and our main disease we study is cancer. And here on these two plots, you can see that the number of cancer deaths in the population, when it is normalized for the growth of the population and for the age, in fact is decreasing. This is US data. You can see that in around 90s there was maximum for many cancers, and now the number of deaths normalized per population is decreasing. Same is true for the world. And this decrease in the cancer death is partly attributed to early detection, but in part to improved treatment. So just to illustrate the same point, this is the probabilities of 5-year survival after diagnosis of specific cancers. For all cancers in compared to 70s, this probability has increased considerably, and then you can see for each specific cancer, there is only one for which there was no increase, slight decrease in probability, but generally, notwithstanding the fact that the number of cancer deaths is growing in every age and in every type of cancer. Once normalized for the aging of population, once normalized for the growth of the population, we are actually having some improvements in cancer therapy and cancer treatment. And why is that? Well, because of the cancer therapy in part, as I said. If I look at the history of cancer chemotherapy, then the earliest chemotherapeutic agents started as essentially warfare poisonous agents like nitrogen mustards. The idea behind these agents was, well, they kill cells, all kinds of cells, but they kill cancer cells a little bit better because cancer cells are faster dividing. And then the whole development went towards discovery of specific drugs. The true anti-cancer chemotherapeutic agents, more or less specific, started from anti-folates in 50s. And then surprisingly, most of the anti-cancer drugs we currently use or currently know were discovered in a relatively short period from 60s to maximum mid 80s or so. So at that time, we saw appearance of multiple drugs like anthracyclines, platinates, and so on and so forth, taxanes, and so on and so forth. Now, more recently, in 90, the antibodies, which would target specific antigen in the cancer cells to suppress the cancer cell growth or suppress the tumor vasculature growth, which is necessary for the growth of the tumor, and so on and so forth. Those antibodies became the clinical reality, entered the therapeutic arsenal. And more recently, we are developing the specific T cells, for example, CAR T cells, cell-based therapies, which would be directed towards cancer cells or towards tumor cells and eliminate the tumor cells more precisely. So you can see that the complexity of our tools is increasing and is shifting from chemistry to biology. On the other hand, the specificity of the tools is also increasing. So as a chemist, I ask myself, are we And when we look at the small molecules, we have a number of problems. I don't want to recite them, but one of the biggest problem is poor solubility of the small molecules. Just to show you one example, this well-known drug, paclitaxel, has been discovered in 1962. So it is as old as me. I was born in 1962. But if you think about it, it took nearly 30 years before this molecule entered therapeutic practice, notwithstanding the fact it has very high activity against many tumor cells. And the one reason for that, and the main reason for that, is that the molecule is extremely poorly soluble. So its solubility is 1 microgram per ml. And only with the discovery of the formulations, which are known as Taxol formulation based on chromophore excipient, as well as more recently Abraxane, only after those discoveries, these very good and powerful drug came into clinical practice. So the solubility is an enormous problem here, and therefore we are looking for the ways how we could deliver safely the poorly soluble small molecules without changing their chemical structure. Obviously, the approach here is to put them into the container, and the containers are have to be nanomaterials so that they can penetrate in relatively dense tumors, and at the same time disperse sufficient amount of the molecules. And there are 2 nanomaterials which have served as the carriers. One is well known, also discovered next year after it was born. It's liposomes. And another is polymeric micelles, and I'm very pleased that I had some impact into the appearance of the polymeric micelles as materials. Working in USSR, we proposed to use pluronic micelles for solubilization of the drugs and delivery them. Originally, we were focusing on the brain delivery, so we were using pluronic block copolymers. We were actually conjugating those block copolymers with the antibodies to the brain-specific antigen. And then we moved towards development of anti-cancer therapeutic incorporated into the polymeric micelles. And the first one was SP1049C. Now it's called differently. It went all the way to the phase 2 clinical trials. And many other micellar drugs have appeared. Most of them thus far were due to the efforts of Kazunori Kataoka, whom you all well know. And the first drug to be approved is Genexol. It was developed by Korean company Semyan Pharmaceuticals. So here's Genexol. It's approved for a variety of cancers, and it contains paclitaxel, that same molecule, which is very poorly soluble. Now, if we compare various paclitaxels in various formulations, I've mentioned 2 of them, the Taxol, the first one, Abraxane, and Genexol. You will see that the drug loading in the Taxol is extremely low. So you have 1% of active ingredient and essentially more than 99% of chromophore and ethanol, which is bringing considerable toxicity during the treatment, and it requires premedication of the patient so you can decrease the immunological side effects, which are often associated with the use of chromophores. So you have excipient dose limited toxicity. Another problem here is quite low stability and low solubility, low concentration of the active ingredient in the final formulation. So Abraxane does a little bit better, and Abraxane is an adduct of the essentially nanoparticles of the drugs covered by human serum albumin. The micellar drug has much higher solubilization capacity and can inject more drugs. So together with Robert and Rainer, about already 10 years or more than 10 years ago, we started collaborating and discovered that the polymers Robert and Rainer were investigating, polyoxazolines, could become a very interesting solubilizing agents for insoluble drugs. So here are the polyoxazolines. I don't need to go into these details. I just want to point out that in terms of the hydrophilicity, the ethyloxazoline is pretty close to polyethylene glycol, which is widely used in pharmaceutical field, while methyloxazoline is more hydrophilic. And both blocks can be, or both polymers can be used as hydrophilic blocks in block copolymer structure. Then butyloxazoline would be an important block here, because that was the first block we discovered to be a very well solubilizing block for paclitaxel. So you can produce tri-block copolymers, di-block copolymers, and in our experience, tri-block copolymers work better than di-block copolymers from the standpoint of stability and formulation. So you have a tri-block copolymer, and you can prepare the micelles. So there are various ways how you can prepare drug-loading micelles. The best way in our practice, so thus far, is blending the polymer and the drug and the common solvent, or melting them together is if melting temperatures are appropriate. Then you obtain the dry powder or a dry thin film, and then you add water and you disperse the system. So the polyoxazoline micelles, when they are right formulated with the appropriate drug, are extremely stable. They are not necessarily spherical, but many of them are spherical. This is a paclitaxel micelles, and they're stable over long period of time. For example, Taxol, also a micellar formulation, the first paclitaxel formulation ever, which was used in clinics. You can see that after several hours, you see a opaque dispersion, precipitation, and this system is, we call it Poxol, with polyoxazoline Taxol, is stable for a long period of time. I'm sorry, I have a dog which is really excited with this lecture, and therefore I have some background noises. Now, just to show you the example, my apology, I need to let the dog out. To show you the example, just comparing the three formulations I was talking about, Taxol, Abraxane, and Poxol, paclitaxel loading is nearly 100 times better, and the concentration is which you can achieve here is extremely high. So now you can have a higher dose of the drug with less excipient, so which allows you, for example, injecting more drug in less volume and with less excipient. Now, if you have less excipient, it is also essentially safer, and that's what we observe. We see improved safety of those formulations, in part due to decrease of the complement activation, in part perhaps due to the overall decrease of the surfactant, which results in more safety. Now, if your injected dose is safer, you can actually inject higher dose with no premedication. If you inject higher dose, you can, of course, achieve higher concentration of the drug in the bloodstream, and you can deliver more drug in the tumor. If you deliver more drug in the tumor, then you can kill the tumor better. And that's exactly what we have seen with high dose therapy, which was enabled with these formulations by injecting our formulations. This is an example of the tumor. I hope you can see my cursor, and it's an aggressive tumor. The animals rapidly die, and the tumor grows very rapidly. it cannot be really controlled at the maximal tolerated dose of regular Taxol. However, Poxol is safe and it completely eliminates the tumor. Here's a comparison with the Abraxane. Again, a little control with half maximal tolerated dose of Abraxane, the complete control of the tumor growth by Poxol. So higher MTD of polyaxazoline-based Micelle pharmaceuticals would allow higher dose therapy and may lead to better treatment outcomes. Importantly, you can use this Micelle technology to easily prepare multi-drug formulations. You do it the same way. You just blend everything together, and now you have the Micelles which contain several drugs. Ability to deliver multiple drugs is necessary. We all know that cancer chemotherapy is using multiple drugs to treat the tumors, in part because in the same cells, different drugs attack different pathways and result in a synergistic action upon the same cancer cells, and in part because the tumor is not just the same cancer cells. In fact, the cancer cells in the tumor is a relatively small fraction of the cells there, and there is a lot of tumor cells which are helping the cell to the cancer cells to grow and constitutes an important part of the tumor, such as tumor-associated macrophages and other cells. So in order to hit the tumor well, you have to address all those different cells in the tumor. Therefore, these chemotherapies using multiple drugs. Now, you can inject multiple drugs in one pill, essentially. From the physical chemical standpoint, what was important that we found here, that we again were able to produce ultra-high loaded formulations of multiple drugs in here. And we did have the loading synergy, so that in some cases we have drugs which are not very well solubilized, but usually when we blend them with a good partner, such as paclitaxel, for example, then we can put a lot of this drug together. So the insolubilization process, drug-drug polymer interactions are at the same time very important. And linked to that, we also have a synergistic retention. So we've seen that the release of the drug was slowed down when we mix good mixing partners in the same polymeric micelle, as opposed to with the single drugs. Now, what does that mean for the pharmaceutical performance? So this is one example. This is a published example again. It's a topazide and cisplatin. It's a hydrophobized cisplatin, so it incorporates into the micelles. We can mix them in a variety of ratios. They are pharmacologically synergistics and used as EPP chemotherapeutic regimen, just separate drugs in clinics. You can see the synergy for the brevity. I just will mention here that synergistic effect can be measured by the combination index. If the combination index is less than 1, then you have synergistic effect of these two drugs. So this is very synergistic combination. Now, there is some interaction by the drugs which relates to the release. Well, generally, the polyaxazoline micelles are holding the drug better than, let's say, regular cremophore micelles. So the drug from the micelle can be released and bound with the serum protein. So we can measure the amount of the serum protein bound drug and the amount of the micellar drug by, for example, by separating them on the extraction column after several, after some time. And what we see here, even in the single micelle system, compared to regular Taxol, this is a cremophore formulation, we see greater retention of paclitaxel in the micelles. Once we blend multiple drugs together, what we observe, this is the topazide and cisplatin mixture, what we observe that for each of the drug, which in the blend, the retention is better than for the same drug in the similar micelle. So there is this drug-drug interaction, and there is a slowdown of release. What does the slowdown of release mean from the standpoint of pharmaceutical performance? Well, we did some modeling and analyzed what could happen depending on the release rate of the drug from the micelles. And to make the long story short, in the case if the micelle is small enough and it can enter the tumor with the drug, then in this case, the decrease of the release rate of the drug from the micelle results in the increase of the exposure of the tumor in increase in AUC area under the curve, measuring exposure, essentially increases the concentration of the drug which gets into the tumor. And that's exactly what we see for the drug blends. Actually, we tried multiple drug blends, and that's a general rule as it appears. So the areas are under the curve, this is the overall exposure for each drug is increasing when this drug is co-blended with a good partner, as it's seen here. So you blend the drug together in one micelle, and these double drug micelle is delivering more of each of the drug to the tumor. Now, if you there are some other pharmaceutical parameters such as blood PK is also improved. So if you put two drugs in one micelle and they are better delivered to the tumor, then the two drug in one micelle will kill the tumor better. And again, that's what we see. This is the blue, or I can see the color, but essentially this is a true drug micelle. You can see that in each case, two drugs in one micelle are either inhibiting the tumor growth and also increasing the lifespan of the animals better than the same drugs administered at the same dose together, but in separate micelles. So that is the effect of the two drugs in one nanocarrier. Now, at this point, I just want to switch the gears and talk a little bit about physical chemistry. It's not my objective to give you a detailed insight in physical chemistry. Very soon, there will be a paper in Advanced Drug Delivery Review published from our group, in which we speak a lot about relation of physical chemistry, polymer chemistry, and key pharmaceutical parameters in the performance of the micellar formulation. I know that Robert mentioned he has a paper on polyoxazolines coming up soon, so perhaps more discussion could be done after reading those papers. But I just want to highlight several things here. One aspect in polymeric micelles which is important is the morphology. And what we've published with Robert, and Reiner, we discovered that the drugs like paclitaxel, they are tending to change the morphology of the micelles from, let's say, non-spherical to spherical. So in some cases, you can prepare, depending on the length of the blocks and so on and so forth, you can prepare non-spherical filament-like micelles, but once you start loading them with the drug, you observe the switch of the morphology to ideal spheres, like you see with the paclitaxel. Now, this is an important transition, and there are several take-home messages here. One is, obviously, there is a strong interaction between the drug, this particular drug, and the core forming block, which results in the change of morphology and perhaps the shell forming block, as I will say in a second. Another aspect, there are implications of the spheres versus filaments in the pharmacokinetics and performance of the drug, which was published in the literature before and which we have now exhaustively studied. And I am not going to show the data because I'm trying not to show unpublished data, but that's an important aspect. And another important aspect here is that we've reported on some drugs which are not shifting the morphology, but which are actually helping to maintain non-spherical morphology of the block copolymer micelles, and the physics underlying this and the chemistry in terms of what's the specificity of the drugs versus given morphologies is quite interesting and important, as I already said. Another aspect from structural standpoint is that, of course, the loading changes the localization of the drug within the micelle, and Robert has published a series of beautiful papers in this regard, demonstrating that for different drugs there is a differences in how they behave. For example, curcumin, as you increase the loading, is tending to shift towards the shell and starts interacting with the shell forming block. And we also realize that it's very important, the choice of the shell forming block. We noticed this early, although Robert has demonstrated very clearly in the most recent publications the importance of methoxazoline versus ethoxazoline. And the bottom line here is the following: as you increase the loading, as you have the micelle stable, let's say at a given loading, and you have spherical micelles, and both are stable with ethoxazoline and methoxazoline. So there is no difference. Apparently, you don't see any difference. Both hydrophilic blocks are holding the micelles in dispersion. But as you increase the drug loading, in the cases when you have interaction of the drug with the corona forming block, there is of course shrinkage of the corona, and the shrinkage of corona of course destabilizes the micelles. So while the polymethoxazoline, being most hydrophilic block, is still holding the micelles in dispersed state, the ethoxazoline does not. That's why the unique properties of our original accidentally discovered block copolymer, tri-block copolymer, butyl-hexazoline with two methyl-hexazolines, are both due to the interactions between the drug and the core forming block, as well as the effects of superhydrophilic polymethyl-hexazoline corona. Now, we've solubilized many drugs, and currently we've tried over 100, I believe, different drugs, and we've noticed that, of course, solubility of these drugs is different in the micelles, and we try to change the core forming block in order to improve the solubility while keeping the methyl-hexazoline block constant. And here, perhaps first major advancement was achieved again by Robert's work. I don't think he was showing that today, but it's already well-known work in which he demonstrated that certain polyoxazines are preferential for certain drugs like curcumin as opposed to polyoxazolines, even though the overall number of carbon atoms in the repeating unit is the same. So essentially, changing the polymer backbone is important for the solubilization in the systems and for differential solubilization in the systems. Now, another study which we've done more recently is we've taken polyoxazolines, and instead of alkyl groups, introduce here triazine rings, and we suddenly discovered that we changed the solubility of the drugs dramatically by doing that. And here, as you can see, we introduce both acceptors and donors of the hydrogen bonds, and the hydrogen bond formation between the drug and the core forming block is important. Now, we've overviewed various ways of trying to predict the solubilization of different drugs in different polymers, and frankly speaking, of all the approaches, perhaps the molecular dynamics with the subsequent computing power can help you. But we also developed a statistical approach recently with Alex Stropsha, in which we use the QSR prediction approach. We're able, based on the sufficiently large training sets, to predict solubilization of the drugs depending on the drug structure. Now, in addition to pure anti-cancer drugs, which are acting upon the cancer cells, we also incorporated a variety of molecules which are changing the tumor microenvironment. You remember in the very beginning, I was talking about the other cells which are there in the tumor, which play important role in the behavior of the tumor and the tumor growth and the development of the disease. So now what we can do, we can blend variety of chemotherapeutic agents and variety of agents which are affecting the tumor microenvironment, those accessory cells which are no less important than cancer cells in the tumor, and we change various biological pathways. Let me give you an idea what I'm talking about. So generally, if you look at the cells present within the tumor, those cells are different from the cells which would be present in the normal tissue. And in particular, there are many cells in the tumor which create immunosuppressive environment for the tumor. Obviously, when the tumor or cancer cell appears, the immune system, and in particular T killer cells, as well as other cells in the tumor, are attacking in the body, are attacking those tumor, trying to eliminate it. However, the tumor has adopted the immunosuppressive cells, such as tumor-associated macrophages, which are polarized to become anti-inflammatory, as well as other cells, which are producing the cytokines, which essentially suppress and kill T killer cells, and this is tumor defense. So the goal of the successful chemotherapy is to shift essentially immunosuppressive tumor to immunopermissive, so our immune system can kill this tumor. To show how it can be accomplished, let me show you one example which we published this summer. Here, using our combination poloxazole and Micelles approach, we were selecting the anti-cancer agents which would be particularly synergistic against lung cancers, non-small cell lung cancer cells. Now, we kind of ran maybe a dozen of combinations, and we determined the combination index. As you know, if combination index is less than 1, this is a strong synergistic effect. So you can see this alkylating agents and cell cycle checkpoint inhibitor has enormously strong, very strong synergy against these cancer cells. This is a combination of alkylating agents with DNA repair inhibitor. Again, very strong anti-cancer effect. And here is just an immunomodulator, which is a TLR7/8 agonist. This immunomodulator has no cytotoxic effect against the cancer cells. What it does, however, it changes by interacting with the TLR receptor 7 and 8, it changes it repolarizes the macrophages from immunosuppressive to pro-inflammatory M1 phenotype. So it doesn't kill the cells, but it just changes the type of immune response. And now this is the animal model. In the real life animal model, our best combinations used at their maximal tolerated dose were absolutely ineffective against this tumor. These combinations, which are highly synergistic, that's because of the tumor microenvironment. They kill the cancer cells very well, but that's not enough. Now, here is the red line, is this is rizikimod. This is the TLR inhibitor in the Micelles alone. As a single drug, you can see a dramatic increase in the lifespan with substantial suppression of the tumor growth. Here, as an example, I'm showing you the treatment with the checkpoint inhibitors. These are the antibodies which are switching off immunosuppressive checkpoint molecules, which are blocking T cells from killing the tumor. For those inhibitors, As you know, recently a Nobel Prize was given in 2018 because they dramatically improved treatment of some tumors, but not all tumors. In particular, in breast cancer or to some extent in lung cancer, the effect of those antibodies is minimal because of immunosuppressive tumor microenvironment. As you can see, the small molecule alone after 4 injections is dramatically increasing the outcome. At that point, I just really want to change the gears, and I will be speaking about coronavirus. Starting March, we were working on coronavirus research, and of course, like hundreds of thousands of scientists across the world. And thus far, in terms of therapy of coronavirus infections and in the therapy of COVID disease, there were few advances. Perhaps the most noticeable advance was with the use of anticoagulation therapy, which decreases the mortality 2 to 4 times in the severe COVID-19 patients. Much effort recently has been devoted to the use of anti-inflammatory therapeutics with little success, although there is some promise with the corticosteroids like dexamethasone. The virus itself causes opposite reaction to what the cancer is doing. It produces the cytokine storm, and therefore trying to mitigate this cytokine storm is one way of treating the more advanced disease. Now, as far as the antiviral compounds, the compounds which would kill the virus reproduction in the cells, well, of course, enormous research was made in the repurposing the compounds, those therapeutics which are out there for various other purposes. And there are several examples. So there are examples to inhibit the transmembrane protease, which is necessary for the entry of the virus into the cells. Of course, much effort has been on the compounds which would block the RNA-dependent RNA polymerase and block the virus reproduction this way. And there were a number of compounds which were discovered which no one knows how they work, but they do work, and at least in the cell screens, and that remains the area of active research in cancer chemotherapeutic. One point about almost all of those compounds, they are very poorly soluble, so the delivery is an issue. And the only compound which was thus far approved for emergency use in the United States is remdesivir, which originally was developed for Ebola virus. And so Gilead has demonstrated in phase 3 trial modest improvement, and mostly improvement to the delay in the disease progression in using remdesivir. This remdesivir is introduced intravenously, either in a 5-day or 1 or 10-day course of therapy at relatively high concentration of 200 milligrams per patient. Now, if we look at this compound, it's extremely poorly soluble, and the current remdesivir format contains 100 milligram of active ingredient and 3 grams with ratio 1 to 30 by weight of cyclodextrin called Captisol. Now, this Captisol is administered at substantial dose. In 5-day therapy, it's 18 grams of Captisol. In 10-day course is 33 grams of Captisol. And in patients with normal renal function, it's okay, but in patients with compromised renal function, it requires the hemodialysis, and nearly half of COVID patients have compromised renal function. So there is an unmet need is increasing drug load and decrease excipient. And obviously, that's the problem which we solve with a variety of our systems. So we have versatility of administration routes here. We can increase the drug content, have high drug concentration. We can administer multiple agents at once, and so on and so forth. So last March, the first thing we've done, of course, we used one of our polymeric technologies, and we produce, well, we actually use several. We produce it through several ways. We produce quite stable, highly loaded particles containing a lot of remdesivir, nearly 44 to 50% loading capacity. We uses 20 to 30 times less excipient than current Gilead's formulation, and which is very stable and lyophilizable. And we believe that these kinds of systems could replace the current formats. Now, obviously, the disease comes to us through the upper respiratory tract, goes to lower respiratory tract, and there is an unmet need to deliver the drugs, antivirals, directly to the respiratory tract. Obviously, if you administer them orally, very little gets to the lungs and nothing gets to the nose. If you administer them IV, the same problem. So we focus on development of aerosolizable formulation, and we very rapidly in April developed aerosolized formulations, which we couldn't use Micelles here because 50% of excipient is too much here. So we, because it would produce some toxicity in the lungs, so we decided to use less than 5% of excipient per active ingredients. And here we developed nanocrystal formulation. Originally we had some foaming issues, but ultimately we developed formulation with no foaming, which contains less than 5%, actually 3% of excipient, 97% of remdesivir, and these formulations are under evaluation. Now, probably we were the first to administer the aerosolized formulation of remdesivir, but already in June 22nd, Gilead developed their own formulation, and declared the clinical trials. And looking forward in the use of our systems towards the antivirals, obviously combination therapy in COVID patients is extremely important. And again, we think that we will capitalize on the same advantages of being able to develop multiple agents at the same time. Thank you for your attention, and I'm sorry I was slowed down a little bit in the very beginning.
Okay, thank you very much.